I guess we are ready for our last microsystem seminar series of the semester. I just want to remind everyone, we have this fantastic seminar series that we launched last year. We have speakers once a month during our academic year, and we have already arranged for a lineup for the fall, so I'm very excited about that. But today is my pleasure and honor to introduce my very good friend, Professor Amy Herr from UC Berkeley. So I'm just going to go over her wonderful bio. Amy received her BS degree from Caltech and an MS and PhD degrees from Stanford in mechanical engineering. At UC Berkeley, her research focuses on instrumentation, innovation to advance quantitation in life sciences and clinical problems. Translational impact of her research programs spans from tools for fundamental research, specifically cell signaling, to near-patient disease diagnostics. Amy, she receives major prestigious awards, including very recently NIH New Inventor Award, the NSF Career Award, Eli Lilly and Co. New Investigator Award in analytical chemistry, and to name but a few. I mean, the list goes on and on and on. She chaired and vice chaired the Gordon Research Conference on the physics and chemistry of microfluidics, has served on technical program committee for various conferences, microcast, Hilton Head, transducers, IEEE sensors, and the list is long, and a guest editor for Lab on the Ship, and very recently, a special edition in the Journal of Micromechanics and Microengineering and Integrated Biology. So it's my honor to introduce her, and she's going to talk about her fantastic research at UC Berkeley. Please.
Thank you. Thank you. Thank you very much for that kind introduction. It's just, I have to say, a great pleasure to be here today, and I've had the opportunity and the honor as well to visit with several people during the day today, and it's just such a lovely place. I have to thank you very much for that, definitely. I'm really impressed and energized as well. So what I'm going to talk about today is work that's going on in my research group called Next Generation Proteomics towards high throughput protein analysis via microfluidic integration. So we work in this area of microfluidics and have for some time. A couple of things I want to point out. I'm an assistant professor in the Department of Bioengineering at UC Berkeley. I also have a faculty scientist appointment at Lawrence Berkeley National Labs, which is just up the hill from our campus in Berkeley. One thing I wanted to point out, which is a little statement that came from National Institutes of Health, technology development is a key ingredient for advancing biomedical research. I would say that many of us sort of accept that as a message from NIH now. A couple of years ago, I would say that wasn't necessarily the case, so I'm happy to see that this aspect of engineering in its all different forms is starting to come into play in terms of this institution in particular. A couple of things I want to point out before I start talking about some of the technical work in my group. We have really two foci of research all towards this idea of bench to bedside. So moving advances and innovations out of the laboratory setting, in our case the engineering lab setting, and into the clinical arena. So that's really the nexus for our work. We work in advanced protein biomarker diagnostics. For the purposes of the talk today, when I say biomarker, I'm referring to a protein biomarker. So that's really our major area of interest. It is connected to and informed by this other area, which I will speak about in some great detail today. And that is really this realization of what I call high throughput proteomics. I don't know if any of you have done proteomic analysis, 2D electrophoresis, mass spectrometry, these types of assays, but they are anything but high throughput as it stands right now. So this terminology is actually an oxymoron. It's a vision that we're working very hard to move towards, but it's something that is important for me to point out to you. So the way that these two major foci work together is these high throughput protein assays that we're developing are really developed towards optimizing the biomarker pipeline process, in particular validation of putative protein biomarkers of disease and dysfunction, which I'll mention in a minute. And the outcome from those assays informs design of the diagnostics for disease prognosis, therapeutic efficacy, and also diagnosis in particular. So we're working really hard in this area of automated quantitative protein assays for more than one physicochemical property. So throughout the talk today, I'll talk about assays that are multi-stage or multi-dimensional, and what I mean by that is the integration of different functions and different readout mechanisms, so that we can really start to dig in and probe this whole protein repertoire that's present in many of the fluids, the clinical fluids that we're looking at. I want to point out that the work that goes on in my group really requires innovation in engineering, and I'm a trained mechanical engineer specifically focused in transport and engineering design, but also leads to our close collaboration with analytical chemists as well as material scientists, and close collaboration with biomedical researchers as well. Typically, clinical researchers and some clinicians as well. So that just kind of gives you a feel. If you're a student who might be interested in this area, we really get an opportunity to work with different people with different technical background, but also with people who approach problems in very different ways. And that's something I've definitely seen in my visit to ISR today. So there's a couple of major projects that are going on. This protein biomarker mining, which I will focus on today, but I wanted to give you a little bit of insight into some of the other areas with a little bit more specificity than I mentioned a moment ago. We have a couple of projects that are looking at proteomics of stem cells, in particular differentiation of stem cells. I won't mention that too much today, but I'm happy to take any questions or ideas that you might have. We're also focusing very heavily in this advanced protein biomarker arena on measurement of protein markers in proximal or compartment fluids. So most of our work doesn't look at systemic fluids like serum, but instead looks at fluids that are proximal to the site of disease or dysfunction. So an example of an area that we're really pushing into is the analysis of human tear film fluid for biomarkers of autoimmune disease. Also looking at viral versus bacterial infections of the eye and being able to differentiate those with very fast diagnostics using that sparingly available fluid. In addition, and this sort of links the two, we're looking at global health diagnostics. We recently developed some homogeneous electrophoretic immunoassays that require separation lengths on the order of about 250 microns. You compare the separation lengths to those that are conventionally needed, which can span millimeters to centimeters, and all of a sudden you're starting talking about running electrophoresis from a battery, a standard battery, and no longer need a high voltage power supply. So you can imagine the implications in terms of untethering electrophoresis from the benchtop and moving it into the field, and in particular implications for global health there. So today, what I'll talk about is protein biomarker mining. So essentially our partnerships with clinical researchers who curate different biomarker sample repositories, so repositories of various human fluids as related to specific diseases. And I'll show you a couple of examples. In many cases, the samples that are collected and stored in these repositories are longitudinal sample collection courses, so essentially being able to take samples from people with a particular disease over a period of time. Those samples generally are sparingly available, so 100 microliters, for example, is a large amount of fluid that's commonly stored in these large, oftentimes -80 freezers, if you will. And our work here is essentially to address a challenge that was posed to me in 2007 when I started this position by a uro-oncologist at Stanford Medical Center who said, I have a patient repository of 50,000 specimens from a prostate cancer study that we started in 1989. I would love to go in and probe each one of those samples for the protein content, and in fact, beyond that, I am interested in different glycoforms of prostate-specific antigen. I don't have the instrumentation, the time, the money, or the person power to make that happen. Can you help me? So that essentially launched us into this area of how can we help her make those measurements in a way that is actually tenable. And that's what I'll talk about today. So when we start looking at this example case of prostate cancer and breast cancer in particular, there's a couple of things that come out. I ripped from the headlines, if you will, the New York Times here recently, a statement from the American Cancer Society, and I've underlined it in blue. This was a major shift in the stance of the American Cancer Society, and that is most cancer screening, the benefits of cancer screening and detecting many cancers, especially breast and prostate, have been overstated. So essentially, the American Cancer Society came out and said, this measurement of free versus total PSA and serum of men, not really working out from the standpoint of prognostic efficacy. Who needs treatment and who doesn't need treatment? It's not really helping us to make that decision. The proposition here is that a large increase in early cancers, meaning that we have effective screening modalities, we're able to detect those cancers very early, would pay off by a decline in late-stage cancers, cancers that are typically untreatable, right? And that makes sense as a hypothesis for what would happen if we had very good screening diagnostics. Happily, with colon and cervical cancer, the methodologies we use to screen for those cancers are quite effective, but unfortunately with prostate and breast cancer, there's this conundrum that's risen recently. this isn't an effective, we have very poor, if you will, screening diagnostics for these two cancers. And the question that we sought to answer is why. So, diagnostic dilemma I have sort of encapsulated here. Prostate cancer screening does not make clear, essentially, which tumors need aggressive treatment and which can essentially be approached with what's termed watchful waiting. So essentially slow-growing cancers or cancers that will never become life-threatening, for example. So being able to identify biomarkers of prostate cancer that would help us to determine the prognostic outcomes for a patient, a particular patient, would be essential in terms of advancing our ability to deal with this disease in particular. What I've highlighted on the left side is a simplified version of what we call the biomarker pipeline. Again, here talking about proteins in particular. We move from a discovery stage, the stage where we're using things like 2D electrophoresis and mass spectrometry to essentially look at patient samples, look at the proteome, basically all of the proteins that are present in those samples, and determine which proteins look different, if you will, between healthy and diseased patients. So which proteins maybe have increased or decreased expression levels, which proteins have post or co-translational modifications, which proteins are maybe complexing in certain disease states. This is what we do with discovery. What I've indicated here is a group of arrows, meaning that there are literally thousands, tens of thousands, if not hundreds of thousands of putative biomarkers that have come out of this process. So if you look in the literature, look at prostate cancer in particular, you start finding paper after paper after paper of biomarkers, proteins that look promising in terms of their ability to help us differentiate disease states, or in this particular example, identify the prognostic outcomes for a particular patient. What's really most frustrating is not the fact that we have all of these promising markers out there, but the fact that very, very few of them are making it into clinical diagnostics. I think there are roughly 20 proteins that are currently used in disease diagnostics, which is astounding, right? When you start thinking about the sheer numbers of proteins that are available, if you will, in a biological living system, and the number of proteins that have come out as likely candidates for inclusion in the screening or prognostic diagnostic. So what is the problem? And we're engineers, and when we look at this pipeline, we essentially say there is several weak spots, but one in particular is the stage called validation. How do we go in and rigorously quantitate whether or not a protein, or more appropriately, a panel of proteins is effective for a particular diagnostic question that we have? How do we, in essence, winnow this candidate pool into something that's not only manageable, but meaningful, right? When we develop our diagnostics, we can engineer the systems, we can engineer the assays, but if we don't know what to measure, all of that work is for naught, right? So that's really the goal of my research group, is to develop better methods for doing validation, to enable, as Professor Peel posed to me in my description of this prostate cancer repository, to enable her to go in and be able to sort through all of those samples that she's painstakingly collected over the years, to point to better ways for diagnosing, and in this case, identifying the prognostic outcome for a patient in particular. So that's what we've been focusing on. Better biomarkers are needed, in particular biomarkers for prognosis with prostate cancer. I mentioned here just the paltry and absolutely stunning lack of inclusion of proteins as new diagnostics. I think I pointed out there's roughly 20 proteins that are used in disease diagnostics today. I want to point out that those 20 proteins don't function for 20 different disease states. I think there's roughly 6 different diseases that those 20 proteins help us to diagnose. So we're not doing so well in terms of development of clinical diagnostics based on protein measurements. So as an engineer, we can look at this problem and sort of dissect it, figure out what the failure modes are. I pointed to this fact that there's a weak link, if you will, at the validation stage. We see, in particular, the introduction of high throughput, high specificity, robust measurement tools for protein measurement as being a way to fill in this weak link. Not only will it allow us to go through those large candidate lists, essentially, of proteins and start winnowing quantitatively what's important in terms of correlation to outcome, but it will also allow us to substantially increase the number of patient samples that are analyzed. That's something I didn't touch upon before and I want to mention now. Typically, when you look at a discovery paper, looking at biomarkers, your sample sizes are tiny. Oftentimes, you'll see an N of 3, an N of 10 for healthy patients and diseased patients. You'll look at a couple of confounding or similar illnesses as well, but we are really limited in our ability to make these measurements at discovery as well as the validation phase. And there's absolutely some people in this room who've been working very hard on that problem as well. So the vision of my group really sort of is inspired by, if you will, it doesn't mimic, but is inspired by this revolution that technology has brought to the genomics area. right? Not only the way we approach genomics questions, but the way we ask genomics questions have changed because of the advent of ultra-high throughput sequencing, next-gen sequencing, all of these different ways to approach these problems. In this particular image, you can see just banks of sample prep and analysis instrumentation that allows us to generate, some would argue, more data than we know how to handle, right? Which is, I think, a better problem potentially than not even having enough data to be able to make conclusions. So our group, my group in particular, is looking at harnessing the power of these microsystems, integrated microsystems, to be able to bring to the proteomics question, this conundrum and these issues, some of the advances that we've seen with genomic science. What I'm going to do today is walk you through one particular example, a suite of examples, if you will, for how these systems, we think, can really start to change the way that we approach proteomic questions. A couple of advantages with microfluidic systems, I have them highlighted here. There's really only two that I'm going to mention. You're probably familiar with the first three that are on this list. These microsystems and some of the fabrication methodologies we are introducing and other groups are introducing are really allowing us to reach levels of integration that are unprecedented in this particular area of technology development, and touching upon unprecedented in the way that we're making these measurements. Okay, so this sort of lends back to the question and the idea that I posed to you earlier for being able to measure more than one physicochemical property for a protein in a complex sample. So we're able to integrate, which lends towards this ability to then automate, right? And free up the time and the resources that are associated with the way we do benchtop biology right now. I don't know if anyone here has ever run a Western blot or 2D electrophoresis. If you could raise your hand, that would be fantastic. Excellent, so just a couple of people. Okay, don't be shy. I know it's painful. So if you've not run these techniques before, you are essentially sitting at a bench in a wet lab, running a slab gel, which can take hours, right, at the very shortest. And you're also coming in the case of Western blotting, looking at the readout from that particular sample and actually physically blotting a piece of material against the slab gel outcome, and then going through some processing that I'll talk about in a minute, staining it and getting a readout, so staining it with antibodies. It is a painful process, it's slow, it requires trained intervention, and it's also not quantitative in most cases. In addition to that, I want to point to the fact that when we start developing these highly integrated bioanalytical systems, we can start thinking not just about mimicking what's being done on the benchtop, but also about introducing new measurement modalities. That's what we're really excited about. Are there novel and important measurement mechanisms that we can start to invent and innovate that will help us to answer some questions that are not commonly addressed right now? So this is the workflow I mentioned to you. For the purposes of this talk, I am going to focus only on our work in protein biomarker mining, so one area that I highlighted, and I've also chosen to show you one example technology innovation that we're introducing. I think it's important to get some depth in terms of the introduction to the technology, and I think this particular case study gives you an example for what types of engineering we have to do to make the measurements we want to make. So I'm going to go in depth here. There's other work going on in my group that I'm happy to talk to you about as well, but it uses some of the same engineering approaches. So, to the Western blot. Essentially what this is a benchtop measurement technique that brings together several different measurements. So I have an example of the workflow indicated right here. Essentially you start off with electrophoresis, a slab gel. So essentially it is a polyacrylamide gel that's about this size. It takes several hours to run and resolve different proteins present in a particular sample. So what you're looking at here schematically, for those of you who have not run this type of separation before, are several different lanes with different samples in each lane. And essentially by using electrophoresis, you're able to resolve different species from each other based upon differences in their mobility or in many cases their molecular weight. So that's the first step. Basically come in and separate all the samples in this proteins in this complex sample. This is called a one-dimensional separation technique. And you can see from the schematic why that is. There's one separation axis from top to bottom on the slab gel. What you do after you get that resolution of the proteins that are present in the sample is essentially transfer to a membrane. So this is that step where you come in with the membrane and physically blot it against the polyacrylamide gel and essentially transfer the protein species to the membrane. You do some blocking and washing steps, and then bring in antibodies that are specific to the protein that you want to measure. So what do you get out of this? You essentially get out both molecular weight or mobility information, so that first physicochemical property that we're interested in. And in addition to that, you also get out interaction or absence of interaction with an immunoaffinity reagent. So that's the second piece of information. And you can correlate those two pieces of information, so we know the molecular weight of the species binding to the antibody of interest, or you know the molecular weight of the species that's not binding to the antibody of interest. So this is a multi-stage assay. Typically, these assays require 5.5 to 22 hours. There's no one in my lab that can run it in 5.5 hours, but those are the numbers that are quoted by the manufacturer, so I thought I'd present that to you here in case you are ultra efficient in the lab and can get that done. There are also upwards of 6 manual steps, so steps where intervention is needed. I've highlighted those with the red arrows here. Something that I didn't highlight is the fact that in order to conduct this one workflow, you need multiple pieces of equipment. You need to have an electrophoresis system, you need to have a blotter or your hands in order to transfer the samples to the membranes, and then obviously you need a fluorescence readout as well as some incubation chambers as well. So we see with this particular workhorse methodology, and I can't stress enough how much this particular methodology, this particular assay format is used in the life sciences. It is around the world in almost, I would say, actually every life sciences laboratory. These types of assays are run every day. We see critical advances here in both speed, automation, and this critical capability to do quantitation. Those are needed, absolutely. I've highlighted the fact that this is a common and workhorse technology for life sciences. I also want to make mention of the fact that it also has dramatic implications for clinical medicine. So after a positive screen test for HIV or hepatitis C or Lyme disease, your sample, a serum sample, is sent to a laboratory medicine facility where a Western blot is run. So if you come back positive, you're sent to sent in for confirmatory diagnosis that has a much higher clinical specificity and sensitivity than the saliva-based HIV screen, for example. So this type of methodology is found the world over in terms of clinical medicine, as well as helping us to address research questions. So what I want to do now is I've introduced you to how these assays are performed and hopefully compels you as to the importance and some of the limitations of, I should say, this very, very powerful measurement mechanism. What I'm going to introduce you to now is the fact that we've essentially been able to develop microsystems that I believe I will compel you dramatically outperform how this important assay is conducted today. So what you're looking at here is a glass microfluidic device. It's about this big on the long dimension. We've added a colored dye here so you can see that it is a combination network of microchannels, and hopefully you can see that in the back, as well as a microchamber, in this case a single microchamber. So essentially what you're looking at is a polyacrylamide gel, so the material that is used for slab gel electrophoresis now. That material has been fabricated with different regions inside the device, so it is a heterogeneous polymer in here, and I'll walk you through the characteristics of those regions, but using photolithography. So instead of using a chemical initiator for the cross-linking process, we use a photo initiator and UV exposure with masking that I'll tell you about in a minute. So let me show you sort of the concept for how this type of assay would work. Essentially, you're looking at that microchamber. I've zoomed in here in this region. I'm going to show you the workflow across the bottom if we were looking at that region. So I have schematically, this is not a real sample. I've introduced this using PowerPoint. Essentially introduce here, over a bright field image of the gels, a plug, essentially, of material from the sample that we're interested in looking at. I apply a potential from the top to the bottom of the chamber, and this defines, essentially, my separation axis. So I perform what's called polyacrylamide gel electrophoresis on this separation axis. In this case, we've introduced a stacking gel, so a discontinuity, if you will, in the gel pore size. You can see that pretty clearly here. We're going from large pore size to small pore size, so when the proteins hit that, they essentially stack, and I'll walk you through the importance for that in a moment. Then we initiate this separation, this polyacrylamide gel electrophoresis. The species separate along the separation axis, and we're able to extract mobility information, electrophoretic mobility information from each of the species as they're migrating. Oftentimes, depending on how we conduct the assay, this mobility information is the molecular weight of the species of interest. So after we do essentially a mini page gel, a micro page gel, if you will, we have more function that's available that's been engineered into the microchamber, and that is the function of being able to now transfer those resolved species to a blotting region. So that step that I talked about before, which happened in the z-axis here, is happening in the y-axis here. So we have those species, we apply a lateral field, we drive those migrating species to the right of the chamber, and what we've done in the far right of the chamber, this region called the blotting region, is included antibodies in that cross-linked gel. So essentially all species migrate to this blotting region. Species that have an affinity for the antibody stop, they're immobilized because of the immune complex formation, and the rest of the species migrate through. Okay, so this chamber essentially represents that separation axis, the capability to do transfer, and the blotting membrane and readout, all sort of unified into one geometry. So in order to realize these, yeah, absolutely.
I'm sorry, can I just ask you all this question?
Yeah.
If you have multiple lanes next to each other, how do you, when you transfer them to the right, how do you prevent the lane to the right of the first lane messing up the measurement?
Yeah. So you can, you're talking about multiplexing,
Okay.
right? Being able to do multiple measurements on multiple samples.
Okay.
We'll talk about a little bit later is a different way to multiplex, but one way to do that is, as you're pointing out, to have parallel separation axes. There are other ways to do it that don't involve these parallel separation axes. But what you're talking about, essentially what we can do is come in and define more than 3 regions in the device. So you can imagine being able to have parallel separation lanes with blotting regions next to them, and maybe you stagger them.
Just one lane.
This is one lane, yeah. There are geometric as well as operational ways to multiplex sample analysis. That's a great point. Yeah, thank you for that question. Okay, so essentially what we had to do to realize something that I've already shown you the results from here, at least conceptually, is introduce some new fabrication approaches. And this is, again, using that photo-initiated photolithography, that photo-initiated cross-linking process. So we have the glass chip. We can come in and essentially pattern the device with several different, actually numerous different gel pore size regions, functional regions. We can do chemical functionalization, biological functionalization, pore size control of the different regions in the device. And I sort of use the term micromosaic here because it kind of gives you sort of physical or a visual idea for what I'm talking about, patterning these little different tiny gel regions inside the device. The way that we've implemented this, and there's a couple of ways to do it, but the way we've reported it on so far is essentially use a very simple multi-stage photo patterning, so mask-based photo patterning process with fluid exchange. Okay, so the beautiful thing about this process is when I have a region of the device, for example here the blotting region that contains a 6% total acrylamide and 3.3% crosslinker with antibody present, in order to fabricate that blotting region, I expose that region to UV but mask the rest of the device. The nice thing about these polyacrylamide precursor solutions is they are very low viscosity. Viscosity is very similar to water. So essentially you can easily flush out that uncross-linked material, introduce a new precursor solution, change your mask alignment, and do a different photopolymerization stage. Okay, so that's a huge advantage because some of these systems that use non-cross-linked polymers, you can get a very high fluidic resistance that requires you to basically apply high pressure to be able to load, like a linear acrylamide, into the device. So that was yes.
What's the typical exposure time?
Yeah, so seconds. Yeah, seconds to minutes. Yeah, absolutely. Depending on the power of your UV source. And being an assistant professor, I could probably get some higher power sources, but what we have now, we've calibrated. Okay. So yeah, and that actually leads me to a point. Thanks for that comment. Essentially what we've introduced and published on is a procedure that, in this case, uses standard life sciences instrumentation. So in this case, a broad spectrum mercury wall. The reason that we did that is, I mentioned that my lab is really motivated by translational engineering, bench to bedside, but as part of that, we see absolutely critically translation of our approaches to other labs, and not just engineering labs, most importantly to life sciences labs. So in designing the process, using standard life sciences equipment, we thought was the best choice from the standpoint of helping to disseminate the approaches and the technologies, and that's detailed in excruciating detail in some of our publications if you're interested in taking a look at that. So again, this multi-stage masking, flushing, polymerization process. I do want to point out that the way that we're incorporating the antibodies in this part of the work that I'm talking about is through a streptavidin biotin linkage. So we include a fraction of streptavidin acrylamide in the precursor solution. So we essentially are left with a blotting region that has streptavidin available for binding. We can co-polymerize with antibody that's biotinylated during that exposure step, or what we've done also, which is actually has pretty big implications from the standpoint of translation, is to fabricate that streptavidin functionalized gel and then use electrophoresis later, so after days of storage, to introduce a biotinylated antibody that can then allow us to electrophoretically pattern the antibody of choice. So we don't have to dial in the antibody identity when we do the photopolymerization. You, for example, could decorate it with whatever antibody you're interested in your lab after fabrication. And that was important from the standpoint of, again, this dissemination, making a vanilla chip, if you will, and allowing people to add whatever flavor of the antibody they're interested in. Yeah, absolutely.
So the addition of the antibody later Is that with pressure or you use?
Great question. Yeah, let me clarify that. So these cross-linked polymers that we're using, these polyacrylamide gels, really support negligible bulk flow. So there is very little electroosmotic flow in the devices, and there really is, I would say, no pressure-driven flow. It's almost like Jell-O. So if you want to try to push fluid through it, there's no way you can. So in the case of the subsequent biostimulated antibody decoration, it is electrophoresis, right? So it must be a charged species. Okay, so what you're left with then is, in this example, a loading region, large pore size gel, separation region, smaller pore size gel. We've done some work on gradient gel fabrication as well, so being able to decrease the pore size as you move down the axis. Some of that has been published for one-dimensional separations. Some of it is currently work underway with these multi-stage assays, and I wanted to share that with you right now. And then you're left with this region of the device that in this case is decorated with that biostimulated antibody through that streptavidin and chromolyte linkage. Okay, so a couple of points I want to bring out that are of great interest to us as engineers, and this is the capability to control precisely the transport that happens inside these devices, the purely electrophoretic transport in this particular system. So there's a couple of weak spots, important areas that I wanted to talk about in terms of designing the control of the system. And those weak spots are highlighted here during the page separation and then the final readout of the blotting region. So there's really one metric that helps me to sort of illustrate the importance of this point, and that metric is something that we call the separation resolution. So our ability essentially to resolve two neighboring peaks from each other, okay? So you might think, wow, if I have two peaks that are spaced very far from each other, so they have a large delta L, I should be able to resolve those two Gaussian concentration distributions just fine. But you have to take into account the width of each of those distributions as well, right? So the variance or the standard deviation. So essentially what separation resolution is it tells you how good your separation is by looking at the displacement between the two peaks, but normalized by the width of those peaks. So I can have two peaks that are very close but have very narrow distribution, and I can resolve them just fine. So that's essentially what the separation resolution is. What I've done here is define two flavors, again, of this separation resolution, one for the electrophoresis stage, this page stage, and then one for the blot as well. What I want to draw your attention to on the right-hand side is I've written this expression in a little different form, a form that accounts for the mobility, the migration capability of each of the species, as well as the average, the length of the axis that we're interested in, here the separation length, if you will, with that L, and then two sources of variance, or what we call dispersion, for those Gaussian concentration distributions. In essence, my Gaussian concentration distribution can broaden owing to diffusion, so that's indicated here with a capital D, or for the case of electrophoresis, it can broaden because of artifacts that are introduced in the injection stage, okay? So that's what this subscript I and J stands for right here. So the way that we approach minimizing injection dispersion in these systems is by using this discontinuity in the gel pore size at the injection. Okay, so I said we have a large pore size gel region, that loading gel region, and in this case we have a uniform pore size throughout. You can see here that I've injected a plug of material. These are inverted epifluorescence images, so the dark regions are the areas that are fluorescing in the sample that we're looking at. And you can see for this particular plug, I essentially have a smear along the separation axis. I can't resolve the various proteins from each other. So I have a very high injection dispersion. Exact same sample, under the exact same conditions, save for the fact that I now have a discontinuity in the pore size right here, inject the plug, and then you can see as those species migrate through and into the smaller pore size region of the separation chamber, we're able to resolve those species from each other. Okay, so I pulled out, sort of quantified here, this metric SR, separation resolution, and you can see that improved resolution just by controlling the architecture of the pore size of the gel in this particular system. So there's a time scale down here at the bottom. Injecting the sample, you can see that porosity here, moves into the chamber, resolves from each other. Looking at the time, less than 30 seconds. This is a 20 to 200 kilodalton protein ladder, so a pretty wide range protein, and even with that uniform separation gel, you're able to see those species resolve from each other. What I also want to point out is when we start looking at the blotting region, we have that injection dispersion source, we have that broadening owing to diffusion, but we have another source of dispersion in the system, and I call that the transfer dispersion. So in essence, I can work very, very hard to get a high resolution separation, and if I'm careless or not rational in terms of how I move those species to the blotting gel and I smear them out, I'm going to lose all of that information I've worked very hard to get in the separation axis. And this is that transfer dispersion. So with the particular design choice that we've made, we are using a chamber geometry, right? Because it gives us several axes for the multi-stage assay. A couple of things to note about these microchamber assays. If you have a chamber where you apply a potential from the top to the bottom, you're going to see field fringing, right? And the material lines in the system, because we are doing purely electrophoretic transport, follow the field lines as well, okay? So when I have fringing of the field lines, I'm going to start to get this blooming phenomena with the charged material that I'm injecting into the system. Likewise, for the injection dimension, when I do transfer, I'm also going to expect to see dispersion that's added to the system because of that field line blooming. So essentially what we've done is come in and introduced an array of side channels, both on the top or bottom, that allows us to inject current, if you will, in the system in order to control and straighten those field lines inside the microchamber. So this was investigated, these approaches were investigated for completely different applications back in the late 1990s, early 2000 timeframe by Bob Austin's group. Some really nice work done by Steve Jacobson's group as well. Again, different applications, but there's a whole idea of correcting the field lines sort of passively, if you will, using the geometric design. So you can see this is some console modeling of those field lines, and in the 2D architecture, we're able to get very nice control. I'd like to show this video because I think it highlights really what's happening. We have the chamber, we introduce fluorescent labeled material in a stream, and then we move that stream almost like a curve across the chamber, right? So you can see we have very fine controlled injection in terms of no field line blooming and fringing, and then also in terms of uniform transfer across the entire axis of the separation chamber. So here, more relevant to our system, we've injected a plug of material. You can see that we're able to inject that plug into this chamber and not see any lateral dispersion associated with movement into the chamber. And then also we're able to move it across the chamber as a cohesive plug of material. So we're not seeing sort of differential migration across this direction of that concentration distribution. So that's important here, and that really highlights our attention to control of this transfer dispersion. Okay, so there's another factor that really requires optimization in this system, and that is how can we get the most efficient blotting possible on the blotting membrane that we're using at the far right of the schematic here. And there's a couple of transport processes that are competing in this particular system, transport and reaction processes that are competing in this system. And we can use a non-dimensional number called the Dompoler number, in this particular case for the on rates, if you will, of the material as it binds to the blotting membrane. Essentially what this non-dimensional parameter allows us to do is to compare the timescales of migration with the timescales of binding, right? So if I move too fast, as compared to the binding, I'm not going to get good binding. If I move too slow, I might not either. Kind of depends on what your system is set up like. In our particular case, there's a couple of knobs that we can turn. We can't do much about the on rate after we select our reagents. That's the antibody-antigen interaction. What we can do is control a couple of things related to how we fabricate the device, so the transfer length scales. The density, this piece event, is the density of binding sites in the material, and how we operate the transfer. So this U, velocity associated with the transfer across the blotting gel. So essentially what we have gone in and done is looking for a range of Dompoler numbers, being able to ascertain how well we're able to capture the material. What is the binding efficiency? And then obviously in this case, we want to get to as close as 1 as possible, meaning that we captured all of the material that's there. So my student Sam Teo was able to develop a simulation using MATLAB and this particular parameter in order to help us identify under what operating conditions we would get that maximum binding efficiency. So you can see that here. What I've done here with experimental results is pull out sort of the timescales that are involved in this transfer, just to show you that these timescales are still very short. Even if we operate at a higher Dompoler number, we're still talking about a transfer that's 30 seconds or so, right? So pretty efficient in terms of transfer to get to those high binding efficiencies. If you're more of the visual thinker, I wanted to bring in some of the data. So this is the chip geometry. This is the blotting region to the right here. If we apply a 50 volt per centimeter field, we see that we capture very efficiently on a small area most of the material that's moving through the blotting region. If we operate at higher velocities, we see that we essentially smear out, but we still capture quite a bit of the material in that blotting region as compared to the length of the region. There's one note that I'll make about this particular system, not just optimizing in terms of capture efficiency, this binding efficiency, but also trying to minimize the surface area where our material is bound. Okay, so essentially trying to enrich or enhance the signal. So I have the same amount of mass or material, and if I can get that bound in a small region, I have a higher signal from that region, as opposed to smearing that same material out over a large region. Similar to an effect you would see with a protein microarray in terms of that detection. So what I'm going to show here, let me just finish this, sorry. So we're doing a separation. We're migrating those species over to the blotting region. I'm just showing you we have two co-migrating species here, one labeled with red and one labeled with green. Obviously, the red labeled protein interacts with the antibody that we've decorated the blotting region with. This is 1 minute and 15 seconds of operation time. Is there a question?
Yeah, I'm sorry, I missed this, but how did you define the efficiency?
Yeah, the efficiency, essentially we are able to use the fluorescent signal. We know how much material we have in the peak, and then we know how much material we have on the blotting region. So we essentially look at the difference between the material.
So how much is captured?
It's how much is captured. Yeah, exactly. Okay, I wanted to sort of tie back now to some of the sort of the overarching theme, the translational or clinical implications of the work. And that really ties to this question that was posed, as I mentioned earlier, about these prostate cancer repositories. So essentially what you're looking at here is the microchamber. We're now looking at a fire color map in terms of the fluorescence information. So the white regions are those fluorescent proteins. And what we've identified here is prostate-specific antigen that's present in a sample that is basically purified human seminal fluid from this repository that I mentioned at Stanford Medical Center. So we're able to do a separation in about 40 seconds and be able to resolve out the PSA, this prostate-specific antigen that's present. We're able to do transfer of this semi-complex sample. It has been pre-processed in terms of decreasing the depth of protein content. But we're able to do that transfer in about 20 seconds, and then we're able to essentially come in and look at which of those bands interacted with the antibody that's present on the blotting gel in terms of our expected interaction with free PSA. Of course, we do the same series of processes using a standard slab gel blotting methodology as well, and also in those samples see this one band interaction with the PSA that's present. So this work is currently moving forward in terms of increasing the separation resolution of the analysis. We've started analyzing lysate samples from cells that express this co-translationally modified form of prostate-specific antigen, and we're starting to run those assays as well. So there's great promise here from the standpoint of being able to capture specifically the marker of interest. I should mention in this case in about 1 minute, which is a great advance over the 5.5 to 22 hours that's necessary for the current state of the art in Western blotting. In addition to that, I want to stress the fact that this integrated system allows us to make this measurement in a completely hands-free manner. So once you program the high voltage power supply and you introduce your sample, you essentially step away from the instrument. And that really starts to lend towards this idea that we have about automation and technology enabling new ways to answer questions and pose questions, not just in the DNA world, but also in the protein world as well. So we're really excited about these types of performance. A couple things I want to point out in terms of directions that we're going. Some of this we published on, some of this we have not yet published on, but I wanted to share it with you today. All of the samples that you've seen so far have been labeled with a fluorophore before the analysis. That's important for us in assay development because it allows us to characterize the transport and the dispersion that's added to the system. I think I've touched upon that in pretty good depth to help justify that. In the samples that we're analyzing from the cell lysates and the patient repository, we don't want to have to label the samples with a fluorophore before we do the analysis. So there's a couple of ways to get around that. In this particular implementation, one way that we have published on is essentially to do the analysis with the proteins that are not labeled and bring in, after the fact, a fluorescently labeled detection antibody. So essentially, in this blotting region, you are doing an ELISA, an enzyme linked immunosorbent or an assay, in this case with a fluorophore. So you have a matched antibody pair. Your readout is, in this case, in the red wavelength range from the fluor that's attached to the second antibody that you're using. Okay, so that allows you to get away from labeling the sample. We oftentimes run the unlabeled samples with a fluorescently labeled ladder that helps us to make sure everything's under control, literally, in the system. There's another really exciting approach that is common in standard biological laboratories, but we think has great potential here for this type of assay, and that is then instead of bringing in an antibody, a second antibody that's labeled with a fluor, bring in an antibody that's labeled with an enzyme amplification system. So HRP, for example, essentially allows us to bring in a substrate and generate product that is fluorescent chemiluminescent on the site of the binding. That, in our hands, has given us about 5 zeptomole sensitivity. So it's a sensitivity, to give you context, it's about 3,000 molecules, which is great for an analytical technique like this, using, again, completely standard life sciences instrumentation. So we're definitely moving forward in that way. A couple things I'm going to touch on in the last couple of minutes that I have. I mentioned this idea of novel assays, and I'm going to move into that space right now. There is a commonly unused methodology, but a powerful methodology that's called reblotting. So essentially you have one sample, you analyze it with one antibody. You want to come in and analyze that same sample with more antibodies, right? So essentially this membrane, we probe it with the antibodies, we then have to strip it and then reprobe it with a second antibody, a third antibody. After about the second antibody, you lose any capability to do quantitation, because in those stripping steps you also lose antigen. Antigen comes off the membrane as well. So what we've introduced here is an approach to do that that sort of marries, if you will, the Western blot with the protein microarray. So here are some of our first results on this approach for the work. But essentially, instead of having one blotting lane, you have multiple blotting lanes. In the example I'm showing you here, we have 2 blotting lanes. We're interested in going up to about 10 blotting lanes. So using this multi-stage fabrication and this chamber geometry approach, 10 is well within reason in terms of what we can fabricate and use in the device. So I've just indicated those here with different colors. So you see page separation, in this case just under a minute, transfer of those species across the chamber, and then blotting of the 2 species of interest in those different blotting regions in a couple of minutes, which is really interesting for some lipform analysis that we're doing. This also lends to some design questions that relate back to how we operate the transfer step, right? And this importance of minimizing that lateral extent of the bound species, not just from the standpoint of enhancing the sensitivity through enrichment, but also confining the species to each strip of antibody binding material. So it kind of gives us design information in terms of what the architecture of this device should look like, specifically these strips of material, which don't need to be the same width, right, depending on the rates of the antibody antigen pair we're interested in looking at. So this is interesting, and we were pretty excited about it because of this capability to make quantitative measurements with multiple probes on one sample. A couple of problems with it. In particular, having to essentially look at matched antibody pairs, so having that decorated polyacrylamide gel with the detection antibody that's present. Having essentially to have questions about how scalable is the approach. I mentioned 10 is sort of the target, but what if we wanted to look at 100 interacting pairs, for example? And I will mention everything I've shown you to date is native protein analysis. So there is no reducing agent. There is no denaturing of the protein before we do the polyacrylamide gel separation. It's important from the standpoint of glycoform analysis because all of those glycoforms are roughly the same size, so they would all move together. Being able to do it under native conditions helps us in some cases to resolve them, but an approach like this also gives us information about them. So we started thinking, how can we do sort of this scalable approach in maybe a more elegant and maybe more functional architecture? So what we decided to do, and this is work that we have not published on yet, is essentially use a surfactant-based system. So SDS, for example, is a common anionic surfactant that's used in protein sizing. So do the separation in the presence of a surfactant. And instead of using antibodies decorating the polyacrylamide gel, let's use charge. So let's harness not antibody-antigen interaction, but let's harness an electrostatic interaction. The beauty of the electrostatic interaction is we can tune it by the charge density in this region of the device, and it will non-specifically capture every anionic protein that is resolved on the separation axis. So I no longer have to go in and sort of decorate this with an antibody that's going to capture one of the peaks. I'm going to capture all of them. And then I'm going to come in and probe with antibodies of interest. So I can easily, on one axis, do 100 antibodies, for example. We've not done that yet, but that's where we're moving. So that's the motivation here. I'm going to show you some of our preliminary data from this to kind of give you a feel for how it differs. So you have that injection junction at the top. You're introducing that fluorescent labeled material. Remember, the blotting membrane now is decorated with a charge. So you're resolving those species from each other. And then we're transferring those species I'm not sure why it's skipped, but you're transferring those species over, and you're retaining all of the species that make it to the blotting membrane right at the interface between the blotting membrane and the separation gel, and that's where that charge is highest. The alpha-actinin in this case is a very large protein, and we haven't given it enough time to transfer over to the blotting region. So this particular system we're using gives us true protein sizing, so not a native assay, and it gives us the ability to capture all of the species from that separation axis onto the blotting axis, which is really exciting. So I'll just make a couple of comments here. You're looking at the page axis. We've essentially gone in and tuned the charge density, so we're increasing the charge density in the blotting gel. And you can see very clearly that we're able to tune our ability to capture in terms of the lateral dimensions of that migrated and then immobilized anionic species. In this particular case, what we've done is come in and tuned the ionic strength of the buffer system. That has impact on the thickness of the electric double layer that's present in the polyacrylamide gel, allowing us to more effectively capture charged species in the case where we have a lot of influence from that interaction. Just in closing, a couple of comments I want to make. I didn't mention earlier our collaboration with a Sjogren's syndrome patient sample repository at UCSF at the Proctor Eye Foundation. But essentially, Sjogren's syndrome is called dry eye disease. It's an autoimmune disorder. It, as you can imagine, leaves the sufferer with very little fluid on the eye. There's very little known about markers of autoimmune disease in general, but dry eye disease and Sjogren's syndrome in particular. So in collaboration with Nancy McNamara at UCSF, we are essentially going through her repository of patient tear film fluid samples and helping to quantify the amounts of some putative markers of dry eye disease, with an eye towards, no pun intended, being able to make a clinical diagnostic that will allow us to, in the clinical setting, differentiate these different types of dry eye disease. The typical progression from initial presenting at the clinic to final diagnosis for a dry eye patient sufferer with Sjogren's syndrome is 6.5 years. That's a quite painful disorder. So essentially what we're doing here is you're looking at analysis of a tear film fluid sample on the blotting membrane. We have an internal standard. We're looking at this cationic protein, lactoferrin in this particular case. We're doing that sandwich assay in a second spectral channel. We are generating dose response information now because, as I mentioned, this is a quantitative approach, but this work is currently underway. I just wanted to mention this other biomarker mining analysis that we're doing. So I think if there's about 5 minutes left. Is that right? Okay, what I'm going to do is actually just skip some of this last information and really highlight here a couple of kind of closing comments that I wanted to make. I think that what I've done with this particular case study is show you how we're able to very rapidly manipulate samples in a very controlled way, okay, by the design of the microsystem, and especially at the integration stage for these different transport mechanisms and separation mechanisms in particular. We're able to pull out multiple physicochemical properties, which is a goal of our lab in terms of these complex biological fluids. Thinking forward, there's a couple of things that I think really stand out from this type of approach. The first one I mentioned right at the start of the presentation, and this is the idea of realizing this high throughput proteomics. Analyses that are quantitative and rapid, but I think most importantly, they're automated, so that we can free up sort of the infrastructure investments in terms of making these measurements. Our particular interest right now is in biomarker validation, so looking at targeted markers and quantitatively vetting or rejecting those markers in terms of their prognostic or diagnostic potential. But I think this has great implications also for the way that we're starting to approach biology in this sort of systems biology approach. What does signaling look like as a dynamic question as opposed to static snapshots? Which lends me towards the final point I want to make, which is the timescales and the automation that are afforded by these systems really start to suggest an approach to proteomics that could potentially lend us real-time information. So what if, from a living system, you could take more than snapshots, but instead get a continuum of expression levels or interactions or modifications? And that's really what we're looking at here, is being able to probe these fleeting dynamic processes that are so key to the types of information that's contained in the biological system. I think microfluidic and nanofluidic methods can really be part of the solution to that. So in closing, I absolutely want to thank my funding sources, my fantastic group at UC Berkeley, as well as the fellowship support that they've received, and fantastic collaborators that I've had a chance to work with. Excuse me. I do want to mention, if you're interested in a postdoc position, if you're a graduate student, I do have a couple available, and I'd love to see you in the Bay Area for sure. Thank you.
excellent seminar. I think we now have time for a bunch of questions. So, who's going to go first? Peter.
Excellent, good.
When you're fabricating the polyacrylamide gel inside these devices from device to device, how repeatable is the pore size you're able to obtain?
Oh, the
when you're
pore size. Okay, I thought you were going to ask about the spatial localization of the different gel elements. So the pore size is very well controlled. So we use commercially available reagents. In fact, we use electrophoretic grade polyacrylamide precursor material. So you just essentially dial in the pore size in a particular systems that I'm describing here by controlling essentially the amount of acrylamide, bisacrylamide, and photoinitiator that you introduce into the system, your UV exposure time. Once you develop a protocol and get a pore size or behavior that you want, you're able to reproduce that really without question. Now, in terms of the actual variation, that's the functional readout, sort of sieving, how well does it perform in terms of reliability. It's very good, I can say qualitatively. In terms of the actual pore size, it's somewhat difficult to measure. There's been a lot of people that have come in and essentially tried to measure it with SEM or different methodologies. One problem with those is you have to dehydrate the material before you measure it, but there have been some really nice work that's been done with that, environmental SEMs too. We haven't really been too concerned with the actual numerical value of the pore size from run to run, but behavior-wise, we see quite reproducible behavior, which is not surprising because, again, this is the material that's used for these types of processes on the macro scale. So a long answer to your question, but I hope that gives you the information you're looking for. Great. Yes, absolutely.
So on the kind of the last project you showed where you were using the charge to capture, so maybe I missed something, but are you essentially just using that to, like, there must be a step in the electric field there that you're stacking against the field, and so it's simply so you don't have to capture with the antibody?
Yeah, so that I actually didn't describe that in great detail. Part of it is because it's not published yet, and also part of it is I was noticing the time.
Okay.
So we'll say what we're essentially doing is using a chemical means to pattern charge inside the polyacrylamide gel. So one common way to do that is to use a group of materials called immobilines that are generally used for isoelectric focusing. So you can essentially tune the charge state based on the pKa of each of those peptides, essentially peptides. And incorporate those in the gel as part of the photopolymerization process. So there is no step in the field. It's essentially a change from material that is neutral, if you will, under most cases, to a material that now has these charges on it.
So you're stacking everything up there. You're not using that to further resolve.
No, absolutely not. Yeah. So the goal there is to let's capture everything, and one way to do that is an electrostatic way. And then let's capture as close as we can to that interface so we get a high signal, and a way to do that is to tune the charge density, in this case to make it pretty high, or change the ionic strength of the buffer. So yeah, let's capture everything and let's probe it later with an antibody. One antibody, not two. So it's more universal.
I wonder if you considered using the focusing technique in the first stage, either IF or ITD or something similar.
Yeah, it's a great question. So we use some equilibrium methodology to get us sort of tight peaks and maybe enhanced mass content, so higher concentration, just concentrating
Better
material.
resolution, higher concentration.
Yeah, absolutely. And in fact, some of the work that I didn't talk about, which is unpublished, is using isoelectric focusing, as a matter of fact, coupled with these tuned polyacrylamide matrices to allow us to do IEF and then immobilize
So
the
you're using
readout.
immobilized gel gradient?
In our case, no. So there's a couple of different projects that are going on. Oftentimes we actually want to move the IEF and then basically pattern the IEF readout on the material, either in one case using light and in another case using charge. So there's a couple different ways to do that, but I love these equilibrium techniques because they do give you a sort of static readout nominally and an enriched signal. One thing that I've been really interested in is actually coupling. I haven't mentioned this to my student yet, so we'll see how she reacts, and you might have some good input on this. But coupling, instead of doing just a transfer of the separation, doing something like isotachophoresis during the transfer to allow us to not change the resolution in the separation axis, but to change sort of the concentration of material as we drive it over the blotting membrane. Because higher concentrations give us better on kinetics for sure. So I think you have a great suggestion.
Very cool.
How does the sensitivity compare to microscopic?
Yeah, so the lower limits of detection. Yeah, so the lower limits of detection for our methodologies I showed for the enzyme linked system is about 3,000 molecules, which is fantastic as compared to like Kamasi stains or silver stains that are conventionally used with Western blotting. Fluorescence-based readouts, I would say that our readouts are on par with what is commonly found with fluorescence-based detection on these 2D gels, for example. That's one way to quantify it. I think there's areas for improvement, for sure, and part of that is the detector. Part of it is manipulation, so enrichment potentially or enhancing the interaction. I think there's improvements that could be made. But presently, with the studies that we're doing in the biomarker mining, we're on par. But we do want to get better, especially for the stem cell biology work that we're doing. Yes?
I'm a little bit unfamiliar with the chemistry, but
Great, yeah.
the last thing that you demonstrated where everything was stacking because of the charges, I'm not sure. Those charges, can you turn them on and off at will?
No.
So it's in the material itself.
Yeah,
okay.
it is. Yeah, absolutely. It is in the material. It's an immobile charge layer that we're essentially adding on top of that matrix, that polyacrylamide matrix. Some of the UV-initiated interactions that we're doing are, you can turn them on just by exposing to UV and essentially allowing the gel to now become active in terms of binding to the proteins that are present. But with the charge patterning, I haven't actually thought about that. I think it's a great suggestion, and the way it's operated right now, no. Yeah, it's a great suggestion.
So actually related, so do you have like a wish list of what technical things you'd like to see better?
Oh,
like would you like to be able to pattern gels better?
The wish list is long.
Yeah, that's a really nice question. I can give you some of my recent proposals.
Okay.
So I think from the standpoint of the performance enhancements that we want to impart in the system, they are all driven by the unmet need or the measurement that we're trying to make.
Okay.
So in the area of the stem cell biology, we have some funding from the WMKEC Foundation to essentially look at single stem cells and try to get at sort of what the secreted proteins are associated with basically muscle regeneration. Why in the fetal stage are we able to generate new functional tissue, but when we're adults and we have a wound, we get a scar, right? So let's look at the signaling that's going on there. A lot of the most interesting nodes in the signaling pathways, I don't think we can detect yet with this approach or with standard approaches either. We can get single cells in, but we don't get that much useful information out. It's all high abundance proteins. So for that particular process, the sensitivity. I think from the standpoint of being able to make some of these measurements in the patient sample repositories, better separation resolution on the separation axis is definitely going to be really, really useful. So those are some of the key kind of example areas. But yeah, good question. Definitely. Great.
Okay, I think with
Thank
that,
you.
let's thank our speaker again. We're going to hang out here for another half hour, so students and postdoctoral, potential postdoctoral, please come up front and talk to her. So before she leaves us.
Oh, yeah.
I remember being honest, just like kidding me. I came back last night. Hello. Yes.
Why don't you look at college education? I haven't heard a single word yet. Not a word.
We'll get it together. Yes, and I know it's already started.
Let me know if at least what I did so far.
I'll try to look at it tonight more
Micro's at 6 a.m.
Yes, exactly. I noticed that you have things.
And what is your position?
So I worked with the
with other people. We really.
Field grade work.
You know her.
So I was
Very good, very good, excellent.
Oh, yeah.
Excellent, wonderful.
Yeah.
Pushing phenomena.
Yeah,
Oh, you were also there? Oh, good, good.
Yeah.